the recording. You have to accept it.
Okay, so now the recording has begun. You should all have your pop-up asking you to accept it. And I am now going to present the plant data management for phenotyping experiment, good practice standards, and use cases for plant scientists. Okay, sorry, I've been muted. So what I'm going to present you has been built over several years by the MyAPI community from several countries, France, Germany, Belgium, UK, etc. And those slide in particular has been built by me, Célia Michotet, and Anne-Françoise Adam-London. So this is a joint presentation between GenResBridge and Agent. So first question, why do we want to have plant phenotyping standards? First obvious thing is to enable anyone, especially yourself, to reuse your dataset. We all had the bad surprise of reopening our old files and just wondering what the hell we wanted to record, to describe in the data we have recorded several years ago. So it's to fetch metadata about the experiment, who did it, for what purpose, where, how, etc. The second incentive to use plant data standards is to enable automatic integration with other types of data, or at least to enable integration in any situation, including by hand. It means, therefore, adding unique identification of the concepts of the things that have been used in your data set. For instance, in a phenotyping experiment, you must be able to link the different measurements, to link the cultivars, the genetic resources used, as well as the location and the timing. And finally, using the right data standards allows you to enable knowledge discovery. Through metadata about the experiment, control vocabularies, ontologies, you are able to make your data visible and to be able to extract knowledge from it. So the scope to which those plant data standards are applied is around genomic, environment, and phenomic. For genetics and genomics, the data is mostly homogeneous. A gene is a gene, whether it's present in Hordeum, in Vitis, or in animals. And furthermore, we have central archives, EBI and CBI, that de facto provide a certain level of standardization. For phenomics, it's highly different. It's highly distributed. It will remain highly distributed. It is highly heterogeneous. There are numerous ways of measuring the same thing at different scale, physical and temporal. So there are a real challenge at the phenomic level. Environment is also heterogeneous and distributed. And we need to be able to bring together all those data to do plant breedings, so to study genetic variations by traits, climate change studies, genotype by environment studies, phenology, etc., etc. And to be able to do this, we need to apply the fair data principles. I think most of you heard about those. They have been published 5 years ago now by Wilkinson et al. The publication is here. And FAIR stands for findable. It means having IDs metadata, so description of your data set, and indexing means to find easily your data or the data of others. Accessible, to have open protocol means to access it, and to have permanent metadata to be sure that in 10 years, 20 years, the description of the dataset is still publicly available. Interoperable, the I of FAIR, means that you have semantics, linked data, vocabularies that allows you to describe the dataset in a way that is understandable by everybody. And finally, reusable, it's first about the license of reuse of your dataset, the legal aspect, to have, of course, the data well described, to have the provenance, and by this, we mean having a good description of the mathematical formulas, for instance, used to transform, to compute your data, and finally, to use the right standards. Those are general principles. The important thing to keep in mind is that there are different levels of applying it. First, by opening your data on the web with an open license, it's the first level of open data. Then, to use a structured format such as Excel rather than PDF, enable the reuse of your data. You know that if you try to copy paste the content of a PDF, it's going to be very complicated. Using a non-proprietary format ensures that your data will be usable in 10 or 20 years. Identified by URIs and relate to other data are more concepts that are very important for computer scientists. What we would like to stress is that this third level, open with non-proprietary format, in this scale that has been built by Tim Berners-Lee some years ago, it's the level that we should aim at minimum, for biological data. So, to summarize, plant phenomics data, they are heterogeneous. There are different spatial scales. You can measure or observe metabolites, organs, group of plants, world experiment fields, etc. You have different time scales with either a single measure or time series with, for instance, measure every 15 minutes. You have the necessity to take into account the environment and to have very precise measure of your environment. And finally, you've got a complex life cycle. Indeed, this life cycle implies to go from raw data. For instance, here you've got a field with a series of microplots. And in each microplot, you've got one accession, one genetic material, soisson, chargeur, etc., and one treatment, high nitrogen or low nitrogen. And from those raw data, from those microplot data, we can generate elaborated data that for a given experiment here allows to say that a given variety under certain treatment has a particular phenotype. It's a mean, it's the application of an agronomical model. So this level of data reduction is necessary to do data analysis, but we need to keep the raw data, and that's very important because this elaborated data has been built for a certain question. If we change the question, we must be able to go back to the raw data to compute it in a different manner. As a consequence, this plant phenotyping life cycle that has been built between the Elixir infrastructure for life science data and the Emphasis infrastructure for plant phenotyping data states that the first step is data acquisition that provides traceability, remeasure, data cleaning, that involve platform information system, that take care of reproducibility of the experiment, of the traceability and the provenance. This data acquisition step is followed by data computation. Once again, for each scientific question, you've got a new computation. Therefore, you've got one raw data set and many computed data sets. And finally, you've got the data publication. You've got one data publication by data set, either for the raw data and for the computed data in fair data repositories. You can say also that you've got a gradient that allows you to go from data, for the raw data, to knowledge. And I think that's the first time I'm saying that raw data long-term conservation is very important, but I cannot stress this enough. So now that I've shown that plant data standards are very important, question is to have a globally accepted data standard. Who is building it? So it's built in an international endeavor based on national network from all across Europe, here France, Germany, but there are also Belgium, UK, Portugal, US, Mexico. In other words, a lot of people are involved in building those standards. They are gathering in European networks, in European infrastructure, elixir and emphasis that I just mentioned. And there is an upper level of global network, such as the CGIARs or the WIT initiative. And all of those groups are building international data standards with the help of the Research Data Alliance. And those I'm going to speak about today are MyAPI. The MCPD is not the subject today, but it's present. The crop ontology, BRAPI is a web service that implements those, but I will not go into detail in that. Once we've got data standards, we must share them. We must outreach. We must promote them. So I would like to draw your attention to standard registries, such as Fair Sharing, which is slowly getting to the position of the reference for standard registries, in which you've got a collection guide of guidelines and standards. Likewise, there is community-driven recommendation list of standards, such as the one built for the WIT-IS, available on witis.org. And the history of how this list of standards has been built is summarized in this paper. So now, this is the history. What are the data standards? There are three categories of data standards. The first one is about semantic. It's about describing your data. It's about building controlled vocabularies, list of terms with their names and their definition, plus possibly semantic link between those terms. And at this level, you go from controlled vocabulary to ontologies. The important thing is that to have efficient ontologies and controlled vocabularies, they must be mostly biologist-driven to really fit the needs of the end users. The second type of data standards are structure standards. They provide means to format and to organize your data. They provide data model to understand it. There are things such as CSV, VCF, GFF, and MyAPI. And to have efficient structure standard, they must be both biologist and computer scientist driven. Finally, something that I will not detail too much today there are technical standards that enables data integration and sharing and provides interoperability means between tools and systems such as GA4GH for genomic data and BRAPI for plant phenotyping data. And those are mostly computer scientist driven. And for all of those to work, one of the most important things is to have persistent unique identifiers, is to have an ambiguous way to identify your accessions, your genes, your traits, but not only at the level of each dataset or of each database, but at the level of the World Wide Web. When you have an accession number, you must have a name to understand exactly which accession is designed by this number. And that's the role of persistent unique identifier, such as DOIs. So, phenotype semantic standards are more precisely ontologies. They describe trait feature in specific plant species, and it's elaborating on the crop ontology model, which is made of a triplet trait method scale. And there is a dedicated presentation and beginning here. The context is that we need to have a data annotation and fairness, and we must capture crop specificities. We must have a very easy way to capture the exact experimental practice of small communities without having to agree globally at the international level. So, for instance, if you've got a study, a genotype, and 3 traits here, SIR, HT, SIR, or here H, SIR, you know that H and HT both relies to plant height, and you must provide a way to link those. This is what the crop ontology provides. It's a list of control vocabularies, which are species specific, and they allow to annotate phenotypes or environment variables. They are community established, and there are currently 31 ontologies crop specific available, including woody plant, wheat, vitis, ordeum, etc. So how it works? As I said, it's a triplet. You've got a phenotyping variable model, which is made of a trait method at scale. Each of those 3, SIR, HT, flow, are a variable. The most important field descriptor for each of those variables are the variable ID and the variable name. SIR is a name. Then we need to add the first element of the triplet, the trait. What is the studied character? Here it's the circumference of the tree. HT is the total height. Flow is the flowering time. Second thing, for a given trait, you can have different way of measuring it. So the circumference is measured at breast height with a graduated ribbon. The height is measured from soil to crown with a pole or a clinometer. The flowering is an interpolation after visual assessment. Finally, you must recall the scale, which can be international system units, centimeters, millimeters, calendar dates, or they can be also notation scale. And I would like to give you now some example of those variables. And for instance, here for the tree circumference trait, you can have 3 different variables: tree circumference at 1 meter, tree circumference, and tree circumference at the collar. The only difference is 3 different measurement methods, but they all relate to the same trait, and they have a clear differentiation. This way, it's much easier to organize the data. Another example I'd like to give is for survival for trees. And I like to use the example of a scoring scale, more precisely a survival scoring scale, in which you attribute a phenotype among 3 possible values, 0, 1, or 2. And in your ontology, you include the definition of each of those categories: 0 alive, 1 dead, and 2 doubtful. Then another example I'd like to give you is the ear emergence in the wheat phenology category. Once again, for the trait ear emergence, you've got 3 different variables. And for instance, you have one variable for the raw data, which is the emergence date. You are simply recording the date at which the emergence occurred. And then you've got the possibility to record variables for your elaborated data, for your computed data. Those are the degree days, which are computed from the raw data. And this can go very far with this final example from the VTIS ontology, in which you see that for the trait flowering, you've got a lot of different variables. You could even have variable to record phenology following the BDCH scale. And once again, you've got raw data with the flowering date, and then computation through degree days, including, and that's important, the formula that has been used here to compute those degree day sum. So this is how we should describe, organize, trait and phenotyping variable for each plant phenotyping dataset. Now, the second question is, how do we organize the dataset as a whole? And this is the goal of the MyAPI, the minimum information about a plant phenotyping experiment. It has been published 3 times now, and the latest paper in the New Phytologist gives a nice overview of the details of MyAPI. So MyAPI is a minimum information for biological and biomedical investigation. The interest of the minimal information approach is that it's mainly biologist friendly. It provides a minimal and sufficient list of metadata, the objective of the experiment, who contributed to the experiment, authorship is very important, what were the experimental procedures, methodology, what was the biological material experimented. So MyAPI has a website, myapi.org. It has been built by many stakeholders, Elixir, European infrastructure, Emphasis, another European infrastructure, Bioversity from the CGIARs, North American PPN, linked to Emphasis, has also contributed. It's a very open community which has requests for comment, GitHub feature requests, mailing list, meetings, work groups, and it's still progressing and it's still getting improved. And it addresses the needs of both crops and woody plants. And to give you a good insight of how MyAPI can be used to capture important information about phenotyping experiments, I like to use 2 examples for illustration. The first one on maize that has been taken from Millet et al. So the OI is here. And the second one from poplar that has been taken from Monklus and al. So for millet, it's a paper, a dataset about a panel of more than 200 maize hybrids that has been experimented under 2 different cultural practices, 2 water regimes, irrigated or drain-fed. It's a drought experiment. It's located in 7 fields, and the temporal distribution is over 2 years. As a result, there are 29 experiments with different cultural practice, different treatments, and repetitions. There is a detailed environment characterization, and you've got a list of procedures. The grain yield has been adjusted at 15% May grain moisture, etc., etc. Those are definitely candidates for crop ontology phenotyping variable. The Monklus et al. Dataset is about 3 field trials located in 3 locations: France, Italy, UK. The biological material are more than 300 genetic resources, accessions. There are good description of how the trial was established, the cultural practice applied, to set up the trial. And finally, once again, the methodology of measure, the phenotyping variable, are extensively described. As a consequence, we can simply consider that we've got, for Monclure, 6 fields, 6 groups of plants. Each group of plants corresponds to one data file. So those are randomized blocks. Each of those points are a single tree, a single thing under observation or observation unit, which has its biological material identification. And on this, we apply the organization of MyAPI, which is based on the ISA backbone. ISA stands for Investigation, Study, Assay. It's an organization of the data that is widely used in life science data standards. Here, the investigation is your whole data set with all of your location on all of your experiments. The second level are the studies. One study is one experiment in one location for 1 to several years. 1 to several because for maize, you've got one experiment by year, one study by year. But of course, for poplar, you've got one experiment over several years, perennial plants. And finally, you've got assays, which are a level of observation, a single plant or a group of plants, for instance, a trait, a method, scale unit, scrap ontology. As I said, the level can be plant, microplot, block, or trial. And finally, the data file is linked, related directly to the assay, and through the assay to the study. So the data file content, as I said that in the introduction, plant phenotyping is highly heterogeneous. Therefore, MyAPI doesn't put any constraint on the format of the data file. It can be near infrared spectrum, it can be images, it can be linked to image archives, etc. Most of the time, we end up with tabular data. And we apply metadata on each column either. We've got accession number. We must have metadata to understand what this accession number is about. We've got trial site. We've got the different measure, height, shoots, etc. And I will describe how it is done. So the investigation, there are research programs with defined aims. They can exist at various scale, grant-funded programs of work with various published components, or they can be a single experiment. One investigation holds one too many studies. Once again, it's important to keep that in mind. And the metadata about one investigation, it's similar to what we provide when we deposit an archive. It's a DOI, title, the description, associated publication, list of authors on their role, etc. For instance, investigation for maize, it's a whole set of multilocal and pluriannual phenotyping experiments. For the poplar dataset, it's a whole set of multilocal measurements over 3 years. Studies. A study or an experiment in the UNESCO semantics on nomenclature. One study is one location. It comprises a series of assays or measurements undertaken to answer a particular biological question. The metadata of a given study, it's the experiment as a whole, it's applied to the experiment as a whole, and it includes timing, location, statistical design, cultural practices, etc. By timing, we mean when does the experiment start and when does it end. Examples of studies for maize dataset, we've got 37 studies, which are a combination of year, location, treatment. For instance, Gaillac, which is a location, 2012, rain. Gaillac 2013, water. For the poplar, you've got 3 locations over 1 to several years. Ardon 2003, Ardon 2003 to several 5, because you can have measure spanning over several years. So to get back to our initial data file that we can see here, you can see that the metadata of the popular study, which are handled in those set of columns, are linked directly to the study level and listed here in this table. The biological material. You are mostly gene bank managers or related, so you know how important it is. So the good thing is that I don't have to stress that today. That's good. It's crucial for integrating phenotyping data with genomic or genetic data. It's a minimal fields taken from multi-crop passport descriptors, the MCPD standard. We haven't reinvented the wheel. We have picked the relevant fields and we link to MCPD and gene banks. And although it includes GPS location for forest trees in situ material provenance. And it has 3 levels: the source of the material used, in other words, the accession, the biological material used in the study, the seed load, the actual plant that is being observed in your study, and finally, samples taken from a given plant. For instance, if you want to do molecular phenotyping, measuring metabolites, concentrations, things like that. And once again, this accession number from your data file is linked to your biological material, which handles all the identification, description, synonyms of your plant material. Following session is the assay level. Conceptually, an observed variable, an assay, it's specific measurements, it targets a trait, it has a method, a scale, and importantly, a trait can be a phenotype or an environment. You can measure soil property in a given microplot through the crop ontology model. So it's a crop ontology trait dictionary trait method scale. And for instance, in each study of millet and R, at the plant level, you've got 20 variables. And at the genotype study level, so a mean statistical transformation from the plant level to each genotype, to each accession, you've got 19 variables, including both phenotypes and environment. At the plot level, you've got 9 specific variables, and it can be, for instance, female flowering days to silking in degree days, plant height, things like that. And in our data file, the observed variables, the assay, are all the following columns. The next thing we need to define in MyAP is the observation unit and the sample. Observation units are objects in the study. Observations, measures, all of your actual data are made on observation units. Treatments, cultural practices, treatment values are also applied, made on observation units. And they can also be used for environmental variables, as I said, soil properties in a given microplot. The metadata of the observation units are specific to MyAP. They include identifiers, location, replication, treatments. A sample is different from an observation unit. It's a portion of plant or tissue extracted from an observation unit. It allows to do subplant observation or molecular studies. And the metadata of a sample includes identifiers, information about processing, extraction, etc. And in our initial example taken from Monku and all, each individual plant is an observation unit. And from each of those observation units, from those trees, we can take, for instance, one leaf and extract 6 leaf discs from each leaf, and therefore we have a set of samples taken from the plant, the observation units. There are other important sections in the MyAPI data model, including events to trace cultural practices, experimental factor, which are the treatments applied, watered, not watered, high nitrogen, low nitrogen, things like that. The experimental factor include biotic or abiotic factors. Its effects are evaluated in that study, and it can take different values in the observation units. It includes as metadata, name, description, and value. As an example, for the maize dataset, you've got 2 experimental factors, treatments, which are rain-fed or watered, and they are identified by dedicated metadata. They are traced through block organization, and they allow to study the effect of environment and growth on the biological material. Here you've got an example. The events are discrete occurrence at a particular time. They can be natural, for instance, rain, unwanted pathogen attack. They can allow to trace cultural practices, planting, watering, etc. They can be applied to the whole study level or for individual observation units. It's not the studied factor, it's an additional information. And the metadata are very simple. It's the name, the description, and the time of occurrence. For instance, in the poplar exemplar dataset, the field establishment date is 2003. The orchard was subjected to 15 millimeters of rain on March 2012. And you've got an example here of how it can be recorded. Finally, you've got environment parameters. They are constant through the study. They don't change between observations units or assays. There are environment characteristics that vary over time. For instance, no, they are not environment characteristics that vary over time. Those environment variables should be recorded as observed variable. Rain, for instance, should be recorded as an observed variable. The metadata are name or value, and some examples are the sowing density, the rooting medium composition, etc. Sorry. So, to summarize, what we can say is that by working on the different level of plant data standards, semantic for crop ontology, structure from MyAPI, you ensure that your dataset are truly understandable and reusable by everybody, including yourself. And you give to the other researchers, to the researching community, and to the computer scientists, the possibility to automate processing of your data through technical standards that will enable you to plug analyzing environments such as the one we are seeing here. Standardize, it's good for you, it's good for others. It's getting more and more easier, and we have been working very hard those past years to make your life easier. And it's an open process, and every contribution is welcome. And I'd like to thank everybody who contributed to this. I am done.
Okay,
Maria,
thank you. So there are 2 questions on the chat from Rick. Van Zede. When do you expect the next version of MyAPI to be released, and what are the improvements or changes compared to the 1.1?
So that's a very good question. I'm not fully sure, and that's something that should be discussed with Emphasis. 2 years ago, we considered that the next version of MyAPI should address specificities related to environment, to traceability, especially for handling different levels. I don't know exactly when this should be done and how Emphasis wish to advance on that. This is a discussion I should revive. It is likely that there will be another minor version meanwhile, because there are some fields that are needed. I am thinking about biological material, synonym ideas that can easily be included. For the environment aspect of Emphasis, we should revise the discussion and see what are exactly the shortcomings of the current version of MyAPI. Maybe they are less important than we initially considered.
Okay, and the second question of Rick was, do you or somebody else know if the upcoming FEES release will incorporate MyAPI aspects, for instance, as an ontology, with the proposed data models or IDs in place?
So MyAPI has incorporated some elements from FEES. For instance, the events are taken from FEES. Furthermore, FEES, which is a database developed by Emphasis France and proposed to many stakeholders of Emphasis, FEES implements the breeding API that I haven't described here, and the breeding API is an implementation of MyAPI. So, to a big extent, FEES is already MyAPI compliant.
There is another question from Sandra. Would you consider an experiment on the same biological material in the same location over 2 years, 1 or 2 studies?
Interesting. It depends if it's perennial plant or annual plant. If it's annual plant, it's different individuals. Therefore, I would say it's 2 different studies. If it's a perennial plant, it depends. I mean, you could have experimental studies that allows you to record your observation over time. Very good. And then on top of those experimental studies, you could do analysis studies that take information from your experimental study, group it differently depending on the analysis purpose that you have in mind. Therefore, on a given plant could belong to one study for the experiment and to another study for the analysis needs. Is it acceptably clear without drawing?
Sandra, did you understand
Yes.
the question?
Good.
There is the answer.
No, thank you. Yeah, that makes sense. Yes, it's I mean, it's clear that in a way, just to understand how you can link the basically the repetitions of an experiment, basically, with these within these studies and within the overall investigation. Thanks.
Yeah, if it's about linking repetition, linking different levels, if you have a series of plot observation, then a series of genotypic mean, I would strongly advocate for putting everything in the same investigation and to have a good traceability to state how you have transformed your data from your plot to your genotype or accession level.
Okay, are there other questions? I'm trying to go through.
Okay, so
there is a new question from Rick about MyAPI.
Yeah, I'm trying to display the chat correctly. Okay. Okay, so in MyAPI 1.1, there is a variable ID, and this variable ID can hold the URI. It is a way to store and to link to the variable as it is created in fields or as it is created in the crop ontology. In MyAPI, what we needed to do is to accommodate with two great categories of use cases. On the one hand, groups having either crop ontology or fields that have a very good definition of their observation variable identified through ID or URIs. That fits in MyAPI through the variable ID, the trait ID, etc. And on the other hand, we needed also to take into account historical data, groups that don't have a database such as fields, less studied species that don't have a crop ontology available for their species, or that don't have a variable suited for their need in the existing crop ontology. And for those, we should at least stress that they must describe the variable, the trait, the method, the scale, and they should have all three of them, not just plant height measured the usual way. And this is why there is a lot of free text available. But if you are using fields or crop ontology, you put a variable ID and you've got all the description in the ontology. Does that answer your question, Rick? Good.
Are there other questions?
Yeah, so those are the screenshot shared.
But yeah. Okay. Okay, if not, I think we can thank you all for your participation and the discussion. And we can perhaps stop there. No question, no regrets. Okay.
Okay, thanks for the webinar.
Yeah, thank you all. And I wish you a very good Eastern weekend.